transgene, apa gene yang kita nak introduce dalam plant tu, dan ada juga gene yang confer resistant towards, for example, kanamycin ke antibiotik lah. Okay? Sebab kita nak select nanti. Betul tak? Kita nak select yang mana transformance. Betul Okay, so similar dengan proses of arabidopsis ni, dia tak perlu tissue culture. Macam kita hari tu, bila kita dah incubate the leaf explant, okay, kita culture on MS medium. MS medium tu adalah medium untuk tissue culture, right? Sebab kita nak kalau explant leaf segments yang being transformed, okay, dia akan regenerate to transgenic plant lah, betul Alright? But sekarang ni untuk arabidopsis, dia ada advantage. You tak perlu pakai tissue culture system. Okay, you cuma perlu ada arabidopsis yang dah berbunga. Okay, that's why kita panggil floral dipping. Dah berbunga. Okay, you celupkan bunga tu, plant ni, dalam agrobacterium yang ada bawa plasmid yang for example, ada gene yang you want to introduce dalam arabidopsis. Okay, ataupun ada segment of DNA. Okay, to create mutants. Okay, then you dip je. Okay, so you allow the plant ni yang you dah dip ni untuk hasilkan seed. Okay, so you collect the seed. Okay, so then you screen the seed yang mana being transformed. Okay, so katakanlah dalam vector ni ada resistance to kanamycin, you patut grow the seed on medium containing ni lah, kanamycin lah. So yang grow dalam medium tu adalah the transformants. Faham
Faham, kiranya dia sekali je lah doktor.
Sekali apa?
Sekali dip je lah. Lepas tu kita dapat apa yang
okay, saya tak pernah buat, that's a good question lah. Saya tak tahu dia dip, saya rasa dia dip sekali je tapi mungkin ada duration lah. So that dia akan infect lah, okay? Alright?
Faham.
Okay. Alright, so can we take a break for 5 minutes? Okay, alright. Okay, kita continue sikit boleh? Lepas tu, tolong ingatkan saya kita nak discuss result hari tujal tu. Saya takut awak tak faham nanti kan apa bendanya kan? Boleh? Nampak ke? Nampak saya?
Nampak.
Okay. Alright, so kita discuss about the genome structure lah, of organisation lah, of arabidopsis. Okay, so this is a paper lah yang talk about it was published in Nature, the journal Nature. Okay, where they reported about the results on genome sequence of arabidopsis in 2000. Okay, so it's actually the composed size is around 125 megabases, ada 140 lebih kurang lah. Okay, so it's one of the smallest genome in the plant kingdom compared to, for example, zinase. The genome size is like 45 times larger. Okay, and tadi saya mention the nuclear DNA is actually carried by 5 pairs of small chromosome. Okay, and then kita dah mention juga the completed sequencing of arabidopsis actually happened in the year 2000. Okay, alright. So this is comparing genetic lah. Okay, so kita cakap kan bila kita study ni, kita study arabidopsis, so the information yang kita dapat daripada arabidopsis, for example the sequences, okay, the genome sequence semua, kita compare dengan plant lain. Okay, so the geneticists have taken advantage of different genetic strains of arabidopsis from different natural isolates, from different ecotypes or from different accessions. And we know that arabidopsis is self-pollinating plant. So each of these isolate derived strains represent a nearly pure inbred line, which is homozygous . Okay, so dalam arabidopsis research juga, there are 3 strains yang kita selalu guna untuk study lah, untuk plant biology. The first one, what we call as the Columbia strain lah. Okay, and then we have the Landsberg erecta strain and also WS strain. Okay, so these strains actually differ from each other, not only in terms of the phenotypes, but also they differ in a significant number of the DNA polymorphisms. Okay, which is the sequence lah of the DNA tu, they differ also. Okay, among the strains. Okay, so different strains also have different alleles at many different DNA marker loci. Okay, so using this characteristic, okay, researchers use this to follow the segregation, okay, of chromosomal regions, which is carrying the markers in the progeny. Okay, so the scientists also can analyze the linkage between DNA markers, okay, as well as between DNA markers and also morphological loci. Okay, so researchers can create DNA marker and phenotypic maps, okay, using this data, okay, and they can integrate the 2 types of map from all 5 chromosomes. So this is, for example, physical mapping of chromosome number 1. Okay, so they boleh map lah. Okay, so the distance between the gene and so on. Okay, you dah belajar kan? I think genetic mapping belajar dekat dengan Dr. Latif kan, betul kan? Linkages semua belajar dengan Dr. Latif kan, betul kan? Okay, so you tahu lah. Okay, the more distance the gene, the recombination lagi happen lagi higher lah kan. Alright, so I think Dr. Latif dah explain lah. Okay, so in terms of the genome, okay, in arabidopsis, okay, dia small size kan, kita dah stress banyak kali dia small size. Okay, because in arabidopsis, there is less repetitive DNA compared to other angiosperms. Okay, less repetitive DNA maknanya katakanlah you ada sequence of DNA, for example, the sequence of DNA ATGC CCA. Okay, ATGC CCA ni the sequence dalam genome of arabidopsis ni berlaku sikit. Okay, for example dalam genome, okay, contoh ini hypothetical bukan betul Okay, katakanlah the segment of ATGCCA dalam genome tu, dalam genome arabidopsis can be found only 6 kali. Okay, alright. Compared to other angiosperm, for example, ATG, CCA dijumpai dalam genome of other angiosperm 50 kali. Okay, so because of less repetitive DNA dalam arabidopsis, it contribute to the small size. Dipaham Ada soalan tak?
Faham.
Okay. Alright, so dia ada less repetitive DNA compared to other genes. And for one gene, okay, kita expect for every 4.5 kb of the genome, you boleh jumpa satu gene. Okay, so this is actually unusually tight arrangement lah. Okay, so in most plant, vast majority of the genome actually consists of repetitive DNA. That's why most plant, dia punya size of the genome bigger. And more than 60% of the proteins in arabidopsis are actually encoded by more than one gene. Okay, katakanlah you ada satu protein A dalam arabidopsis. This protein A di-encode by more than one gene. Maknanya ada gene 1 and gene 2. Okay, can produce protein A. Okay, right? So this is example. Alright, and analysis of large duplication in the genome suggests that actually arabidopsis ni derive daripada ancient tetraploid. Okay, so the arabidopsis ni derive daripada ancient tetraploid, tapi arabidopsis adalah diploid. Okay. Alright, so this observation suggests that a large extent of functional redundancy in arabidopsis. Okay. And like other species of plant, arabidopsis genome contains multiple copies of transposable elements. Okay, and I believe Dr. Latif also dah explain about transposable elements. Okay, apa transposable elements ni? Belajarkan transposon kan? Belajar ke?
Belajar.
Apa transposon?
Yang berpindah-pindah tu.
Okay, segment of DNA that can be transposed ataupun boleh jump from one location from another location, alright, in the genome. Okay, good, alright. Okay, siapa yang answer my question tu?
Nuri.
Okay, thank you Nuri. Okay, so arabidopsis ni genome ada transport element lah. Okay, alright, and also dalam arabidopsis dia ada juga the genome, nuclear genome ni pieces of DNA yang originate from mitochondrial and also chloroplast genome. Okay, alright, and as similar to animals, the centromeres of the chromosome are heterochromatic, which is rich in repetitive DNA. Okay, and the genetic and physical distances vary widely along the plant chromosome. Okay. Alright, so this is sebenarnya kan, okay, ni data yang saya kata daripada paper published in 2000. Okay, during that time, okay, so dia classifykan arabidopsis genes ni based on the function. Okay, based on kategori lah. Ini paper 2000, okay. Sekarang ni dah banyak yang unclassified ni. Sekarang ni dah 2022, 22 tahun yang lepas, unclassified ni banyak dah being classified. Okay, saya nak cakap, saya nak stress yang ini adalah data 2000. Okay, alright, so the biggest category of genes yang being identified contribute to the metabolism of the plant of arabidopsis. Okay, followed by transcription and so on lah. Okay, just to show you the function of the genes based on category. Alright, tadi kita dah cakap the importance of transformation. Okay, sekarang kita nak cakap the importance of mutant screens. Okay, based on the mutant, kita dapat tahulah, okay, the genes yang contribute to the phenotype. Okay, so mutant screens play important role, okay, in the emergence of arabidopsis as a model genetic organism. Okay, so kita dah mention banyak kali the short life cycle, small, efficient production through self-pollination. Okay, so it can so untuk study mutation senang. Okay, so in the late 1950s and 1960s, okay, these scientists actually use mutagenic treatments, okay, to create mutants. Okay, so dia treat embryo tu dengan mutagen, okay, so that to create mutants, which the mutants is actually deficient in chlorophyll, or the mutants are actually sterile. Okay, so the arabidopsis, okay. And the earliest ni kan yang involved dalam mutation ni adalah they treat the arabidopsis with EMS. Okay, so EMS ni adalah agent, adalah chemical yang cause mutation ya. Okay, so EMS ni adalah agent yang paling famous diguna masa awal-awal. Okay, but they still remain effective until today. Okay, untuk create mutants. Okay, so bila you want to create mutants, you treat arabidopsis tu, okay, the seed ke, the embryo ke, you expose, okay, dengan EMS, okay, then kita akan dapat mutants lah, okay. Okay, so using this mutants ni dia ada variety of phenotypic. Okay, so for example, deficient in chlorophyll lah yang paling famous lah. Okay, the most prevalent. Okay, and then the suitability of arabidopsis for biochemical genetics was confirmed through thymine auxotroph. Okay, which the seed actually with a little phenotype lah. Okay, so ada banyak lagi lah. Okay, you boleh baca ni. Saya just nak tunjuk contoh-contoh Okay, techniques ni kan saya tak nak discuss dulu sebab kita akan ada satu topik, remind me to go back to here, okay, untuk discuss balik about techniques untuk create mutants. Ada banyak techniques nanti kita akan belajar in detail. Okay. Alright, so this is how kita create mutant, okay, using agrobacterium. Okay, bukan create mutant, kita nak study the function of the gene, kita develop using promoter of gas, okay, alright, and then okay, this example of mutation, mutant, okay, you can see on the left ni adalah mutant actually, and the right, sorry, the right is actually mutant, okay, and this is the wild type, okay. Alright, so the cotyledons on the mutant is actually you nampak abnormally expanded compared to the wild type. Okay, alright. Okay, so this is another mutant. Okay, this involves mutation of the gene GAL1. Okay, which cause dwarfism. Okay, so this is the wild type arabidopsis yang normal. This is the mutant lah. Okay, this GAL1 kita dapat tahu dia involved in dwarfism. Okay, so this is also another mutant plants. Okay, where they involve an exposure of ethylene exposure. Okay, so this is the wild type. Okay, comparing to mutant when they exposed to ethylene. Okay, so what happened to the seedling? Okay, so this is comparison. Okay, so this is another mutation, okay, which is developed in response to light. Okay, so ni kita akan belajar in detail juga later. Alright. Okay, so any question? Okay, tak ada. So arabidopsis ni. Okay, so I want to show you a video. The next topic will be comparative genetics. Okay, so I want to show you video first before I explain about comparative genetics.
Chinese speaking preferred. How many of you face this when you're sick and tired of your current job?
What is comparative genomics? Comparative genomics is an exciting field of research in which scientists use a variety of tools, including computer-based analysis, to compare genes or the complete genome sequences of different species. By carefully comparing characteristics that define various organisms, including the genomes of organisms ranging from humans to chimpanzees to yeast, researchers can pinpoint regions of similarity and difference. This information can help scientists better understand the structure and function of genes, which is extremely important. Identifying DNA sequences that have been conserved, that is, preserved in many different organisms over millions of years, is an important step towards understanding the genome itself. It helps identify genes that are essential to life and highlights genomic signals that control gene function across many species. Comparing genomes can eventually help us to further understand the networks of genes that are associated with certain diseases, which might lead to the production of more effective medication. Comparative genomics is also a powerful tool for studying evolution. By analyzing the evolutionary relationships between species and the corresponding differences in their DNA, scientists can better understand how the appearance, behavior, biology, and biochemistry of living organisms have changed over time. Comparing genomes also allows to figure out the evolutionary relationships between species and construct trees to represent them. As the DNA sequencing technology becomes more powerful and less expensive, comparative genomics is finding wider applications in agriculture, biotechnology, and zoology as a tool to identify the often subtle differences between species. Such efforts have led to new insights into some branches of the evolutionary tree, as well as improving food production and proposing new strategies for conserving rare and endangered species.
Okay, so that is comparative genetic. Nama pun compare kan? Okay, right? Alright, so I will give you the link nanti dekat ayat link. So nama comparative genetics. So what is the meaning of comparative genetics? Okay, so the comparative genetics is really where we use the information from the sequence genome of an organism, okay, to relate the organism to other organism, okay, without the sequence genome. Okay, so it's called comparative genetics. Okay, so sekarang kita dah tahu arabidopsis fully being sequenced. Okay, alright, so tapi kita ada jumpa satu plant yang dia punya genome tak sequence lagi. Okay, jadi bila kita extract the DNA, okay, of the plant yang kita tak tahu, yang dia punya genome belum completely sequence atau tak sequence lagi, apa tu kita compare dengan sequence of arabidopsis. Okay, so by doing this, kita dapat tahulah, okay, for example, kita kata, okay, this is identify, mungkin gene ni, bila kita compare dengan arabidopsis, for example, adalah involved in flower development. Okay, so this is what we say comparative genetics. So we use the information from the sequence genome, for example, from arabidopsis, to relate the organism to other organism. Okay, alright. So large-scale sequencing project like whole genome sequencing can be extremely costly. Sebab kalau kita nak sequence semua kan, okay, dia actually akan jadi, sebenarnya sequencing ni dulu compare dengan sekarang ni dah lebih murah lah. Tapi actually nak sequence semua plant dalam universe ni actually costly lah. So that's why not many big genomes have been sequenced. So this is why we said that comparative genetic is valuable. Okay, when you want to assess gene for important traits that have been identified in the sequence genome, but not in genome of other important organisms. Okay, so regarding both human in arabidopsis, so the sequence genome of arabidopsis can be made to compare with other plants. Okay, so for a human, okay, can be made to other animals. So for example, tadi nampak kan dia tunjuk human sequence and so on. Okay, so for example, okay, if kita locate identical disease-associated region in other animals like mice and chimpanzees, researchers can use this organism to glean insight to human gene function. Okay, so kalau kita rasa region ni of the genome ni, actually this region involved in disease, okay, in animals, for example dalam mice, okay, dan kita boleh deduce this information to obtain information that kita kata this region also in human involved in this disease. Okay, so that is contoh lah. Okay, so the benefit of comparative genetics is actually banyak lah. For example, time saving, flexibility and low cost lah gene bank. Okay, alright, okay, and also there is also benefit and possibility to compare genetics in animals also attain with plants lah. Okay, so kita cakap tadi arabidopsis is very important lah in comparative genetics. Okay, so far banyaklah arabidopsis telah digunakan untuk compare dengan rice, tomato, corn, broccoli. Okay, for example, arabidopsis homologs or identical genes have been found in rice, for example, and function similarly in both plants. Okay, so these two scientists use comparative genetics to compare between 9 different grasses. Okay, so when they studied, they found that many genes and their order on the chromosome were relatively same for all these 9 species of grasses. Okay, so they concluded that in the future, grasses with small genome, for example like rice, can be used to study grasses with much larger genomes, for example, like corn, without having to sequence the entire corn genome. Okay, so this is the advantage lah of comparative genetics. Okay, ada soalan? Tak ada?
Tak ada.
Alright, good. Okay, so kita move to green revolution lah. Okay, so kalau you adalah plant science, okay, students, plant scientist, kalau you tak tahu Grand Revolution, memang you nak kena ketuk lah, okay. So, bila orang cakap Grand Revolution, you akan automatically relate dengan this scientist yang initiate the Grand Revolution, which is Norman Borlaug lah, okay. So, Norman Borlaug actually initiated the Green Revolution, which is a movement, okay, to produce higher yielding crops, okay, in response to the population explosion, okay, and also the reduction in farmland occurring worldwide. Kita tahu kan the number of population in the world keep increasing, okay, and also with the modernization, okay, area untuk farmland being reduced lah. Okay, so example yang famous, high quality traits of crop, okay, from wheat and corn to peas was selected lah during this generation using classical breeding and innovative methods, okay, to increase the yield lah. Okay, so plant breeders look for traits that would make certain plants more productive in grain or fruits. Okay, so this is we call as ni lah sebagai green revolution. Okay, alright. Alright, the selected varieties were successful in producing more grains and fruit than previously coveted varieties, but they were drawbacks lah. Okay, for example, drawbacks, the plant, the more productive plants required large amount usage of chemical, okay, to enhance the size of grain or fruits. Okay, chemicals, for example, fertilizer is expensive and also toxic. Okay, so farmers in poor country had to invest in substantial amount of money lah to buy fertilizers. Okay, and by selecting plants for large fruit trees and removing small fruit trees with other type of genes, the plant breeders actually sometimes remove the important genes lah needed for other plant functions like defense against disease. Okay, so in 1970s, okay, which kita akan belajar lagi juga later, which is CMS, a corn variety called Texas milk steroid had been bred, okay, for traits ranging from high yield and harvesting ability to disease resistance. Okay, but not all the disease resistant genes, however, had been bred into the Texas milk steroid. This is the Texas milk steroid vulnerable to unimportant pathogens lah. Okay, so in fact, one such pathogen, which is Helmintosporium midis, which causes the southern leaf blight disease, attacked the cornfield in the United States. Okay, which were actually in the US, they plant the 90% of Texas milk steroid lah. Okay, so this disease eliminated half of the corn crops. Okay, so bila dia nak dapatkan higher yield, okay, bila dia select some of the traits tu, dia akan menyebabkan dia jadi resistant, prone to diseases lah, okay, to attack to diseases. But luckily, some other corn varieties have been safe. Okay, so they quickly being planted and reduce the effect of the devastation. Okay, so that's all. Alright, any question? Okay, saya dah letak lecture notes dalam iTaklim. Okay, any more questions? Ada?
No.
Okay, can we discuss before kita stop about the experiment yang you buat genomic DNA extraction? Saya takut ada yang tak faham lah Alright. Sekejap Okay. Hello. Sorry. Okay, kita nak discuss result hari tu so that nanti you faham apa you nak tulis report dia. Okay, so saya letak bila you kena hantar lab report tu. Bila you kena hantar lab report, saya letak 20 Mei kan? Okay, hantar 20 Mei Okay, so kita tunjuk, kita tengok result kita. Lama, saya terlupa nak letak dekat. Saya letak ke? Yang bajau tu? Dekat e-taklim boleh siapa check? Saya rasa saya letak. Okay, so choose papers to read. Okay, lab. Okay, so kita ada buat dengan saya, ia ada buat lab genetic transformation. Okay, lab 2 genomic DNA ni Okay, so alright, so saya ada letak ni submission of lab 2 you nampak Okay. Okay, which is due date. Okay, it should be submitted on 20 May pukul 11.59 malam. Okay, and dekat sana saya ada tulis the lab report will be individual assignment. Okay, so okay, dia akan based on introduction, 2 markah, objective, very clear, result and discussion and conclusion. Okay. Alright, so saya diletakkan ni, kita tengok gambar ni. Alright, so these are the gambar. Boleh nampak ke?
Boleh.
Alright, so the first lane dengan the last lane kita letak ladder. Okay. And then kita letak lepas tu sampel awak lah G1, group 2, group 3 and so on. Dan hari tu you dah measure the concentration and also the purity Okay, so 1 kb ladder ni sebagai ni dia adalah, okay, dengar baik-baik saya explain. Ladder ni untuk ni sebenarnya kita nak ukur size selalunya. Tapi in this case, okay, 1 kb ladder, kita tengok saya tunjuk kat website. Kejap. Hello, Ladder Roslan. Salam. Okay, nampak dekat ladder ni, okay, dia akan nampak band. Okay, so band ni yang paling bawah, macam saya katakan, DNA ni bergerak berdasarkan kalau dia punya saiz lebih besar, okay, dia akan bergerak lagi lambat. Okay, kalau dia saiz dia smaller, dia akan bergerak ni. So, you nampak yang paling bawah, the size of the band is 250 base pair. Okay, saiz ke atasnya 500 and so on. Yang paling besar adalah 10,000 base pair. Okay, tapi yang kita extract ada, okay, you tengok sini nampak nampak ke?
Nampak.
Okay, so yang kita nampak sekarang ni, and kita tahu genom, kita extract genomic DNA, the genome. Okay, macam tadi kita compare, okay, kan kita cakap tadi, arabidopsis pun the size of the genome is berapa tadi? Berapa tadi?
114.
Betul, 140 megabases. 140 megabases. Arabidopsis yang the smallest genome So you bayangkan, you extract ni adalah apa dia? kita extract genomic DNA ni dari apa?
Rupa.
Jatropha, betul. Itu pun dah lupa. Aduh, nasib baik ada orang ingat. Okey, kita extract genomic DNA Jatropha. Jadi the size of genome Jatropha lagi besar daripada the size of arabidopsis. So, tapi the size of the ladder yang paling besar adalah 10,000 base pair. Jadi in this case, okay, sorry, terbuka pula saya punya chat. In this case, ladder kita guna bukan untuk determine size, tapi kita nak tengok sama ada eksperimen kita berjaya atau tak. Okay, so sekarang ni kita nampak dia ada smearing. So you nampaklah semua ni berjaya dapat genomic DNA. Okay, kecuali G4 ni macam tak clear. G9 ni saya nampak ada lah. Okay, so G4 ni mungkin dia punya extraction fail lah. Faham tak maksud saya? Okay, ada question tak?
Kalau yang fail tu kira cakap fail lah doktor?
Fail lah. Dan you kena cakap lah kenapa you fail, apa possibility, betul tak?
Okay.
Alright. So jadi yang lain ni semua kita successfully isolate. So dalam lab report itu you kena discuss lah your result ni, dan you kena discuss concentration dia, you kena letak juga dia punya purity. Macam saya kata, kalau sampel DNA kita pure, the ratio of 280 over 230 tu should be within 1.8 to 2. Okay? Alright, so any question? Saya rasa clear kan? Alright, so okay, kalau apa-apa yang you tak clear, you can always ask me lah. Okay? Alright?
Doktor.
Yeah. Alright. Okay, alright. So petang ni lab dengan Dr. Latif. Kemungkinan saya tak join lah. Okay, so I wish you selamat Hari Raya. Okay, have fun, balik raya, balik kampung, celebrate. Okay, alright. So kalau ada apa salah silap, saya minta maaf. Alright, so we see you in 2 weeks time. Boleh?
Boleh doktor.
Okay, Assalamualaikum. Selamat Raya.
Terima kasih doktor.
Selamat Raya.
Doktor.
Syaifah, you boleh tak WhatsApp saya? Saya kena jumpa dekan lah. Saya boleh entertain, ataupun kalau saya tak dapat jam petang sekarang, you WhatsApp saya boleh tak? Boleh? Sorry, dekan dah call saya nak kena pergi KLC. Boleh tak? Okay, thanks. Tapi jangan lupa email WhatsApp saya. Alright.